prebiotic. Finally, the result in the formation of the simple organic compounds. Now here, the biodegradation of complex molecule of a pesticides into a simple inorganic compound. This type of the transformation is done by the microorganisms. The process is called as bioremediation or biodegradation, whatever it is. Now coming to the biomagnification, as I explained earlier, due to biomagnification, the toxins will be retained in the higher species, leading to the health hazards. That means biomagnification means transfer of toxic or hazardous molecules or chemicals from lower individuals to the higher species or higher individuals through food chain or food web. So my motto is to break or to avoid the biomagnification by biodegradation of these chemical pesticides by using bacteria. Next, now coming to the Food and Agriculture Organization as defined as a term of pesticide, any substance or mixture of substance are used to intend or destroying or controlling the pest. Okay, the soil bacterium capable of utilizing the pesticide as a carbon source. So carbon source was isolated by the selective enrichment on mineral and medium containing the pesticides. Okay, medium along with the pesticide should be used for the preferable for the isolation of the bacterium. The bacterium degraded the pesticide in high concentration, that is 50 to 200 milligrams per liter also. Next, organophosphorus in the pesticide. The main component present in the pesticide is organophosphorus in pesticides such as like parathion and methamidopus, chlorpyrifos, and cypermethylene and chlorantraniliprole. Actually, I'm using the pesticide is a chlorantraniliprole, the group of highly toxic agricultural chemicals widely used in the plant protection. Current theories laws are mainly concerned with the separate aspects of microbiological degradation and the biodegradation of the pesticides in the soil. Actually, sir, this is it is a field. I collected the sample from this field. The sample is collected at tomato field in the Telangana region. Okay, this next. Now coming to materials and methods. Sir, now coming in these materials and methods, the bacteria is isolated from soil by using following techniques: the serum dilution technique and pore spread and secret methods. We all are very well known about this serum dilution and pore spread method. Now coming to the collection of the soil sample. The soil sample is collected from the tomato field. That area belongs to the Gopalpet village, Khammam district, and Telangana. Actually, it is my village, sir. I collected the sample from my field only. The bacteria is isolated from soil by using colony technique. Is a mainly colony technique. From the collected soil sample, the organism isolated through serial dilution, preferably 10 to the power of 6 dilution, was spread onto the nutrient agar plate and incubated. After the incubation, the colony growth was observed. From this, one colony was picked up and streaked onto the again fresh nutrient agar plate and incubated along with the pesticides, and also spread over the plate colonies were observed. Next, so it is a picture of my slides. Next, coming to the spread plate method. These are the pictures are collected from the Google. These are the spread plate images and. Now coming to the structure of the pesticide, that is just I'm using the pesticide is a chlorantraniliprole. It is one of the broad range of the pesticides. Generally nowadays, farmers are continuously using using to the field for protecting their crops. Actually, it is consisting of the benzene ring, pyridine ring, and pyrazole ring. They are interconnected by the amine linkages, along with the halogen. Halogen is attached with the third carbon atom, that is chlorine. Next, so it is a biodegradation pathway of the chlorantraniliprole. Actually, the biodegradation is mainly done by the bacteria. Bacteria consisting of the enzyme is organophosphorus dehydrogenase. The single enzyme it is in multiple forms. It is having the capability to degrade the total amount of the pesticide, highly compact structure of the pesticides into broken into a small fragment. Whenever these small fragments are formed, these fragments are consumed by the bacteria as a sole carbon source, because the bacteria is a exhibiting the chemolithotrophic type of the mode of the nutrition. Next, now coming to the major criteria of my research work is the optimization studies for pesticide degrading bacteria. So in this slide, for optimization studies, that there are the number of parameters are used. In that, the first parameter is effect of temperature, effect of pH, concentration of the substrate, pesticide concentration, and time. These are all the optimization conditions we are maintained artificially in our laboratory, in my laboratory, for successful growth of the after a successful growth of the pesticide degrading bacteria after the isolation. So after the isolation of my bacteria into the from the soil, then this bacteria is has to be purified by the puricultures by selective media is minimal essential media. Minimal essential media is a selective media for my studies.
Here, last 5 minutes. Last 5 minutes.
Okay, sir. Here, optimization conditions. Sir, in this picture clearly shows that before degradation of the sample and after the degradation of the sample. Here, the flask are consisting of the selective media is essential minimal essential media, which consisting of the salts along with the pesticide with the organism. I'm adding the different concentrations of the pesticides like 10 mg per liter, 20 mg per liter, 30 mg per liter, 40 mg per liter, and respectively, followed by I should maintain the different pH conditions 4, 5, 6, 7 respectively, and followed with the different temperatures 25, 35, 45, 55 are the different temperatures, and also added the different substrates like glucose, fructose, sucrose, and lactose. So these are all of the ideal parameters for optimization of my bacteria. So after this, all the type of the optimization conditions are maintained. After the inoculation of this flask with bacteria, with bacteria, the bacteria is having the capability to degrade the pesticides like well. So it is a before degradation. This one is a after degradation. So next results. After the optimization studies, the result predicted is the temperature should be 37 degrees centigrade. In some literatures, the successful degradation of the pesticides in the temperature is 35 to 40. But my strain is having the capability to degrade the pesticide in 37 degrees centigrade, followed by the pH. In this pH, actually the bacteria is isolated from soil. The soil pH is the cultivating soil is the pH should be 6 to 8. But we are in my studies, the pH predicted is as optimization pH is a 7. Okay, the next comes to substrate. So by the usage of the sucrose as a substrate, the bacteria which can uptake the more amount of the sucrose and finally degrade the more amount of the pesticides. Next, pesticide concentration. Sir, pesticide concentration 10 mg per liter is 10 mg per liter is maximum degradation is observed at this stage. Sir. Next, time takes 5 to 30 minutes, 5 hours 30 minutes. Actually, under the ideal conditions like temperature, pH, substrate, and pesticide, the time should not be considered as a parameter. Next, now coming to conclusion. So after these type of the findings, conclusion of my topic is screening of the microorganism that shows the pesticide degradation was done successfully by using selective media as per the literature study. Sir, next, the isolating bacteria is capable to degrade the pesticide in higher concentration at optimization condition. Sir, I am taking the parameter is 10 mg per liter. Complete degradation is observed at 10 mg per liter, but the remaining degradation, that means 50 to 60% and partial degradation is observed at the high concentration of the pesticide, that is 100 mg per liter. Here 30, 40 and 50 mg per liter concentration also be sufficiently degraded by my strains. Next, the optimization of the organism has to be established further at its genus biochemically as well as species molecular level, sir. Actually, it is a further continuation of my research, sir. Just I did up to the my research work onto the screening and optimization of the pesticide degrading bacteria. As his understanding may give a more clear picture about this organism role at molecular level in effective degradation of pesticides in further levels. Thank you.
Okay. Thank you very much and very nice presentation. And now it's open for question
Thank you, sir.
answers. Please, if you have one or two
Okay,
questions,
sir.
please ask to unmute your mic and please ask. It's open to all.
Yes, sir. I'm in line, sir.
Okay. Anyone have questions? Please let me know. Okay, Pawan, I have some questions with you. So see, you mentioned here pH 7 for the soil. pH becomes 6, then the microbe will change, the nature of microbes.
Of course, sir. Sir, actually, we are maintaining the 5, 6, 7, 8 pH, sir. At the 7 pH, the maximum degradation. That means the degradation means the maximum degradation must take place, sir. At the pH of 6 also it is one of the parameters to degrade the bacteria to degrade the pesticide. But when compared than 7, 6 is the minimum.
And is it actually single bacteria or a group of bacteria that they are degrading the pesticide?
Sir. But
like which bacteria you found it? Did you find it out? Which bacteria?
No, sir. Actually, I did my work up to only screening and optimization, sir. Next, I have to establish the species and molecular studies, sir.
Okay. Thank you very much for your presentation, and I thank you very much. And please, good luck for your future work. Please now, stop your sharing.
Okay.
Thank you very much. And now next we have presentation. E. Satish Kumar from Department of Zoology, Presidency College, Chennai. I request you, Dr. E. Satish Kumar, please share your screen. And his title is Studies on the Inhibitions Bioactive Compounds from Segra Water Treatment and Purification. Okay, please start. Oh, yeah, you can see. Please start. Unmute your mic and start, please.
Hello, good morning to all. Yes.
Start, please, sir.
Studies of inhibition bioactive
Yeah, Satish
compounds
Kumar,
from
please
marine
start.
algae. Yeah, studies on inhibition bioactive compound from marine algae Sargassum to water treatment and purification at Department of Zoology, Presidency College, and Research College, East Research Scholar. Yeah, introduction. What is algae? The algae is a group of different organisms from different phylogenetic groups. Algae can be referred to as plant-like organisms, do not have a true root, do not have stems, leaves, vascular tissues, and have a simple reproductive structures. Most of the Indian towns are not sufficiently planned to contain large populations, and in many associations, small towns have really essentially into cities to area settlement rather than plant development. Water is one of the major products of nature used enormously by humans being, and it's not unnatural. Then any growth community generates enormous wastewater of sewage. As a clean environment, it prerequisites for healthy living and any urban settlement. Proper treatment and safe disposal of sewage call for prime attention. Untreated wastewater can cause pollution for surface and groundwaters. Many new developments in the field of sewage treatment are eventually taking place. Attempts in the treatment of effluents contain materials such as phenols, paper mill sludge, distillery wastes, rubber stress wastes, olive oil mills wastes, and heavy metals. Though the treatment domestic wastewater using cyanobacteria was attempt to evaluate the efficiency of marine algae to treat the wastewater under the different growth conditions with a view of develop more efficient technology which save the less expensive technique modifications. Materials and methods. Marine algae collections. The marine brown seaweed Sargassum was collected from Rameswaram, Southeast India, state of Tamil Nadu. Immediately after the collections, the sample was transferred to the laboratory in a new plastic bag containing natural seawater to prevent infection. Wastewater sample was collected from different points. The sample collected were analyzed for their physical chemical properties. Samples for biological analysis were aseptically collected sterile container and their transport immediately to the laboratory and subject to physical, chemical and biological analysis within 3 hours of the collections. Physical characters such as color, odor, temperature, and total solids, and the chemical characters such as pH, dissolved oxygen, biochemical oxygen demand, chemical oxygen demand, hardness, amount of chlorides, phosphate, and sulfate nitrogen was estimated following the analytical procedures. Character of wastewater. Physical. The color of the wastewater is brownish black. Temperature. The temperature of the range of 27 degrees Celsius to 31 degrees Celsius. Low temperature was observed in the month of December. And the temperatures above 99 degrees Celsius were recorded in the month of May. Total solids. In the sample, total solids 8 mg per liter and 192 mg per liter. Chemical action demand variations of COD was appreciable during the study period. A minimum of 310 mg per liter December and the maximum 427 mg per liter. And April was the same 20 mg per liter. Treatment process. The cell column size of 50-50 into 50 centimeters was prepared with the culture of algae, which is inlet and outlet values of the column was picked with the algae. Batch treatment process. The packed bed column was tested for its treatment of water. The column was filled with allowed the remaining such treatment. Total period of 96 hours. At regular interval samples were drawn analyzed for the COD and BOD. Algal stain was also removed with sampling and cell and protein. Continuous treatment. Wastewater was subject to continuous treatment for algae cell located in the packed column. The prepared column was fed wastewater from bottom and top, flow rate 25 mg per hour, 50 mg per hour, and 75 mg per hour using periodic pump. At regular intervals, the sample was drawn over the total period of 5 hours. Reductions of COD and BOD was estimated. Effect of inoculation concept of sargassum, myristerium or crucial treatment of wastewater, exposure rate of percentage reduction. Initial level of COD is 380 mg per liter, BOD 186 mg per liter of normal sewage water. COD incubation period over 48 to 72 90 seconds. The total period into incubation period, incubation concentrations of 5 minutes 15.8 for 48 hours of COD. 10 COD is 16.3, 15 minutes COD was 17.2, 20 grams for 14.5. Is a BOD level also 48 hours 6.5, 10.2, 14.5 and 11.3. Results and discussions. Sargassum shows a maximum growth of municipal wastewater at 300 degrees Celsius, 30 degrees Celsius. Each state results presented in table incubator the 3 cells of this algae, 20 gram inoculation concentration. Without any addition, you could bring about more than 50% reductions of COD BOD after 96 hours. This is 96 hours. The POD COD was reductions. The COD is 49.5. The BOD is 48. For the reductions. Table for the treatment of wastewater cell using cells of sargassum is packed column batch process. Express the term percentage of reductions. COD is 24 hours. Stain type 28.3. BOD is 24.5 of 24 hours. Comparison of various mode of treatment of wastewater and sargassum. Pre-cell treatment of 96 hours. COD is 50.5. And BOD is 52.2. Batch process is 24 hours 35.6 and BOD is 30.1. Continuous process is treatment level 41.2 and BOD is 39.8. Conclusions. It is evident for the table of the continuous treatment of the wastewater. Sargassum is more suitable and ideal since a maximum of 53.1 and 51.2% of COD and BOD reductions could be observed respectively for the 20 ml flow rate treatment. Although the waste marginal decline of 6% of reductions of both the COD and BOD after 5 hours of continuous treatment. Enhanced activity of COD and BOD reductions could be observed with the sarcasomyces cells under continuous treatment compared to the batch process and the pre-cell treatment. The increased activity may be due both the slow movement of wastewater along the column, which could be facilitated better contact and activity by all cells cumulative contributing of the total reductions of COD BOD. Clearly enunciate the potential for sarcasm to express enhanced activity of continuous treatment compared to the static content of the wastewater batch process. The present studies speculate that there is unlimited scope of using such potential strain enhanced activity towards rapid treatment and probable recycle of the wastewater for useful purpose. Thank you.
I think we lost connection. Okay, anyways, now we move to the next one. Next PPT we have from D. Sarvani. She is Assistant Professor of Chemistry at Aditya College of Engineering and Technology. And title is Correlational Analysis of Soil Sediments in Industrial Area of East Godavari Region. I request to D. Sarvani to please share your screen. Is D. Sarvani here? D.